Good morning, everyone. Our primary speaker today is Maha Shady. Maha did her PhD in the bioinformatics and integrative genomics program, or BIG, at Harvard University, and is a member of Eli Van Allen's lab at Dana-Farber. Today, she will give us a primer on how large language model-based agents work. Please join me in welcoming Maha. And yeah.
Good morning, everyone. Thank you very much for having me today. So today I will give a primer on AI agents in biomedical research. And I'm sure many of you have seen that there has been an increasing number of publications and even companies that are working on agentic systems for biomedical applications, whether that's research workflows or for clinical support. And my goal from today is to talk specifically about what is an agentic AI system, what are the components that go into developing this system, and then to spend the second half of the talk talking about evaluation of these systems, especially in the biomedical setting, and key points that are important to keep in mind when designing these systems. So first, what is an agentic AI system? A paper from, or a definition from a recent review describes it as a system comprising one or more computational models that operate autonomously to achieve human-specified goals by processing data to guide solution generation, decision-making, and execution. So typically, in a machine learning model, the model represents a single bounded transformation that takes an input, processes it through a single forward pass in order to generate an output. That could be a prediction, an embedding, or perhaps a generated image. And the decision for when and how to use the model relies on external logic. But in an agentic system, the model participates in an iterative decision loop where a context is assembled and provided to the model. This can be a user prompt or results from prior iterations through this loop. The model then reasons over this context and generates an output, which could be an answer or an action to be executed. And if there's an action, then the result of that is then observed and appended to the context for future iterations through the loop. And two key distinctions here are one, that the model participates in deciding what happens next, and the degree of model autonomy versus human intervention is a design choice that the developer implements when designing the system. And the second is that having this context that accumulates across iterations through the loop can influence future reasoning steps. And so this makes the model more powerful in executing long-term or complex tasks, but it also means that there is a possibility for error propagation or cascading of failures. So now I want to discuss some key algorithms that make base models powerful for such agentic systems. The first, as you might expect, are large language models. So these are transformer-based architectures that are trained on large corpora of text and code in order to perform next token prediction in a self-supervised manner. And training on large amounts of diverse data leads to the emergence of broad knowledge, reasoning capacity, and natural language communication within these models. And essentially, they become the reasoning and language backbone of the system. The second is supervised fine-tuning, in which these pre-trained LLMs are curated on prompt response examples in order to help with the emergence of desired behavior patterns, such as instruction following, reasoning traces, and tool call formatting. And here, data quality is really important. And these prompt response examples may be generated by humans or through strong teacher models. And finally, reinforcement learning can be used in order to help shape behavior patterns that are difficult to learn by imitation alone. So this may include reinforcement learning through human feedback, where humans can compare outputs and train a reward model that can be used to help update the policy. And this could enforce features like helpfulness and alignment with human values. The second can also be reinforcement learning with verifiable rewards. And so this is where a binary reward is given on tasks that are easily verifiable. It can help enforce behavior that more likely will lead to a correct answer, such as, for example, extended reasoning for harder tasks. And in this reinforcement learning step, a KL penalty can be used in order to make sure the policy does not drift too far from what was learned during the supervised fine-tuning step. So now, what are the components of these systems? First, there's the base model that we've been discussing. This is the reasoning backbone of the system, and it can also engage in things like task planning and reflection on past behaviors. And in a single iteration, it simply observes a prompt shown here through the context, and then produces an output. But in an agentic system, there is a loop where the LLM produces an output that can become part of the updated context for the next step through the loop until a final output is produced. And as I mentioned, the context includes the user prompt. So this can be a particular question or a goal for the agent to work towards. And it can also receive any relevant data or document for performing the task. And everything is wrapped in a scaffolding. So this is code that is used by the developer in order to basically manage the entire loop. So it helps assemble the context, pass information between system components, as well as perform tool executions. So an additional important augmentation is what I just mentioned, which is the ability to execute tools. And this is given to the system usually by specifying particular tools the system would have access to, such as web search, or the ability to run code or query databases. And the model can then generate a structured output representing a particular tool call, which is processed through the scaffolding and fed back to the context through the next loop iteration. And finally, an important part, especially for complex long-term workflows, can be memory management. And so typically we'll see some partitioning into short-term and long-term memory. And these memory stores are able to essentially read and write from the context, and we'll discuss them more later in the presentation. You can also have multi-agent systems. So in this diagram, I'm abstracting away the context box just to make things a little bit simpler. But in this setting, you can essentially have multiple LLMs, which represent specialized sub-agents that perform particular tasks. And you can also have an orchestrator agent, which performs goal decomposition and coordinates between the sub-agents. So now that we kind of gave an overview about what components go into the system, I'd like to get into more detail on each one. The first thing I'd like to talk about is the reasoning of the base LLM. So one concept you may have heard about often is chain of thought reasoning. And this concept was formalized by this paper from 2022, where the authors showed that prompting the LLM to provide reasoning before giving a final answer in a few-shot manner produces correct results more often. And this helps enable tackling complex problems where standard prompting may not have very good performance. And they demonstrated this on multiple types of tasks, including arithmetic problems like the one seen here, as well as common sense problems. So these are problems that require reasoning over physical interactions or human interactions, as well as symbolic reasoning problems where the agent needs to manipulate abstract symbols according to a particular set of rules. And the key insight here is that the more tokens spent reasoning about the problem provided correlates with better performance on hard tasks for sufficiently large models. And you can also enforce more complex reasoning, which might help with tasks that may benefit from considering multiple potential reasoning traces. One example is through self-consistency, where multiple reasoning paths might be initiated, and then majority vote is used to select the final answer. And the key insight here is that correct reasoning path will converge onto the same answer. And you can also have something such as tree of thought, where reasoning paths are iteratively evaluated, pruned, and expanded until a final answer is reached. And so in such a setting, different search algorithms can be used to traverse the tree. And particularly in the paper, they demonstrated breadth-first search, which allows exploring multiple promising paths at once until one reaches the desired objective, or depth-first search, where the most promising path is explored until either the objective is reached or there is a failure, in which case you backtrack to the parent node and then consider the next most promising path. And again, as I mentioned, these methods can help with tasks that may benefit from considering multiple reasoning traces. But of course, as the complexity increases, the computational cost also increases. And so this is an important trade-off to keep in mind. So how do you actually enforce reasoning traces into your system? The first is by using a reasoning model as your base LLM. So these are the models that I initially described that were trained with supervised fine-tuning and reinforcement learning in order to encourage reasoning traces. Additionally, you can enforce it through the scaffolding code itself, potentially by requiring a structured output format that includes a thinking block and then reprompting the LLM if it does not produce such a block, And you may also implement specific reasoning mechanisms such as tree of thought in the scaffolding. And finally, you can also utilize the system prompt, for example, instructing the model to think step by step before answering, or providing few-shot examples of the desired response format, which includes a thinking block. And one important note to keep in mind here is that while reasoning traces might help the model reach the right answer and might also be helpful in overseeing what the agent is doing, it is not always a faithful representation of how the model actually produced the answer or selected an action. So this paper by Anthropic actually showed low faithfulness scores across different model types. And they showed that this faithfulness decreased even further on harder problems. And so overall, this says that the reasoning trace, while helpful, is not sufficient for monitoring unintended or unsafe behaviors. And then we've mentioned actions multiple times here, and this is where the tool calls come in. And so I'd like to talk more about that. In the think, act, observe loop that we've been mentioning, this is essentially the paradigm that was introduced in the React paper. And the act part refers to tool utilization by the agent. So how do you actually give the agent ability to utilize tools? First is, again, through the base model that you use itself. So many frontier models have native function calling abilities. And so these were trained using supervised fine-tuning in order to enable the model to parse JSON schemas that describe available tools along with their required arguments. And then these models are then able to generate structured output with valid tool calls that can be parsed in the scaffolding. And this generalizes to tools that were not necessarily seen during training. And reinforcement learning can also be used here to help shape behavior around tool calls. For example, when is it appropriate to call a particular tool versus just answer directly based on reasoning? And then the second is, again, through the scaffolding. So this is where the structured output from the model is processed and the tool is executed, and then the result is appended to the context window for the model to observe through the next iteration through the loop. And finally, the system prompt can also be helpful, such as demonstrating through few-shot formats the types of tool call formatting that you're looking for. And this becomes particularly important if you're using models that don't have native function calling abilities, or if you want to use a format different from the JSON schemas that I just described. So one alternative format for tool execution was provided in the Kodak paper, where essentially the agent provides action as blocks of Python code, which the scaffolding then executes in a sandbox interpreter. And so here the agent can include multi-step logic in a single action, and it may also self-debug by observing the Python traceback that is fed back through the scaffolding. And in such a setting, additional functionalities such as, for example, web search or database queries can be wrapped into functions and made available by the developer so that the model may call these functions as part of the code blocks. And two LLM capabilities that we've kind of alluded to and I'd like to discuss more are reflection and planning. So in terms of reflection, it would be beneficial for the agent to use that observation part of the loop in order to help give feedback to update or modify its behavior. So this might help it accumulate experience over time, as well as potentially avoid the propagation of errors. And one paradigm that was proposed for doing this was in the reflection paper, where an evaluator component, this can be an LLM or a heuristic function, generates a score for the context trajectory thus far. And then a reflection agent uses the score along with the trajectory to generate verbal feedback that the actor LM then observes and can use it to modify its behavior. And this verbal feedback can also be stored in long-term persistent memory in order to help kind of accumulate experience. Alternative strategies may also include utilizing external verification into the reflection process, for example, by inserting checkpoints for human feedback in the loop, or through tool use as done by Critic. And so here the model interleaves calls to external tools, which may include, for example, search engines or code interpreters, in order to verify claims in the model's reasoning and also identify potential errors in the output before generating a critique that is then given to the model. So this essentially helps ground the reflection in external evidence rather than only relying on the model's own reasoning. And in biomedical setting, you might think of various ways to kind of try to enforce reflection into your system. One can be, as I mentioned, by just having checkpoints throughout the loop for human feedback that can be then fed back to the model. It can also be kind of implicitly included in the React loop because the tool results themselves can serve as external grounding to revise reasoning if the output, for example, returns an error or contradicts a prior claim or hypothesis. And one example of this can be the utilization of vision language models to assess figures that the agent generates and make sure kind of they are consistent with the goal and with the reasoning so far, and this can help provide feedback as well. And then you can also encourage it in the system prompt by instructing the model to revise its reasoning or showing few-shot examples of the model correcting itself. And then finally, planning can also be an important capability for multitask or multi-step workflows. And one example of how to do this was shown by Hugging GPT paper, where an LLM controller is prompted to create a plan decomposing a user goal into subtasks. And then accordingly, it selects models from Hugging Face that can execute each of these subtasks. Once these are executed, then the language model synthesizes these outputs in order to provide a coherent response to the user. And in biomedical applications, this may look like simply a multi-agent system where an orchestrator agent decomposes a task and delegates individual steps to specialized subagents. So the main missing piece that we haven't discussed so far has been memory management. And as I mentioned, you'll often see the separation into short-term and long-term memory. So short-term memory refers to the context window that's being assembled by the scaffolding at each loop, at each iteration through the loop, and passed back to the LLM. And this may include the user prompt.
Can I ask a question on the previous section?
Sure, yeah.
Just to make it more concrete. So let's say you use like something really silly, simple, like use an AI agent to do your bulk RNA-seq analysis, and then you want to put that in your paper. When you write the methods section, what do you say about how you verify the outputs of that work? Like, what do you actually have to write there to make somebody believe that it didn't make some silly mistake somewhere? Like for me, I feel like I would still have to look at every single line of code. Is that the standard? Or is that what people should be doing? Because like, you can make all these little heuristics and tests, and I talk to my RA and I ask, did this gene come up? Or, did you see this? These sanity checks, but I don't really have to sanity check them for doing crazy things usually. Yep. But I still feel like that's a little on the table for some of these. So what would be like when you're writing your methods and say you use an agent to do something, like going back to this critique part and the thinking and self-reflection, like what would you have to write there?
Personally, what I would do is actually check everything that the agent does. So then, really the utilization there is that it helps kind of speed up the process by initiating the, the RNA-seq analysis. But personally, I would check everything in order to make sure it's okay, so talking a little bit about short-term memory. As I mentioned, this is just the context that's being fed to the LLM at each stage. And it can include the user prompt as well as the history of reasoning traces and results so far. And it is session specific and also limited by a finite token ceiling. And so it may need to be managed as the session progresses in order to make room for new reasoning. And different ways to manage this may include, for example, prompting the model to generate kind of working notes, which may include key insights and decisions made so far to be included in the context at each step. And so in that sense, if later on in the session, the scaffolding removes earlier parts of the history, you kind of avoid the issue of potentially losing really important information from that part of the history. Additionally, you might also use token level compression of the context in order to optimize storage. So this is, for example, was shown with LLM Lingua, where they used a small language model to prune less important tokens at every iteration in order to optimize usage. And then long-term memory refers to external persistent stores, and these offer more flexibility in terms of length and the types of information that you can store. And one paper proposed viewing the different types of information that the model might need to store in long-term memory through the lens of cognitive science by dividing them into three different types. The first is episodic memory, which essentially includes information on past behaviors, and this may include a summary of past runs and failures that can inform future attempts. The second is semantic, so this refers to facts and knowledge and may include, for example, vector index documents as used in RAG or databases that the model can query. Although database queries actually practically represent tool calls, they can still be viewed as a form of long-term memory because they represent information that the model can access as needed. And finally, you can have procedural memory, which includes rules that influence the agent's behavior. So for example, the system prompt and tool schemas can be considered a form of this memory, as they are stored in files that the agent can access as needed. And although technically they're also included in short-term memory, as they're always part of the context at each iteration, their availability in external stores and persistence across sessions makes them also a form of long-term memory. And you may also have additional protocols that describe how the agent should behave. So sometimes you might see these being referred to as agent skills. And these may include, for example, summary of QC best practices or data processing pipelines that the model can access as needed. So now that we've gone through details of the agentic AI systems, I want to talk a little bit about evaluation of these systems, especially in the context of biomedical applications. To start, evaluation of these systems is difficult. So, in real world tasks, you may not always have a single correct answers. And in fact, if you're actually approaching a novel research task or an unseen clinical case, you may not know what the answer is. And so this makes performance very difficult to measure. And also, biomedical workflows include diverse multi-step tasks that are difficult to evaluate through Q&A benchmarks alone. And finally, as we just discussed in the question actually that was asked, evaluation of procedure and not just results can be really important because you want to make sure that the model is following a sound workflow and there are no security violations, for example, such as the leaking of sensitive data or performing actions without oversight. So now I'll go through a few examples of particular benchmarks that attempt to measure capabilities that are important in the biomedical setting. The first I'm going to talk about is SWE-bench or software engineering bench, which measures agent coding abilities in real world settings. And this is a core competency for agents in a biomedical application setting. So here the authors put together a set of open source Python repositories that have posted issues, a solution, as well as test cases for the provided solution. And the models were asked to edit the code base in order to resolve the issue. And then they were evaluated by whether or not the edited code passes the associated tests. And actually, in this initial evaluation, they observed low performance where the best performing model solved about 2% of the tasks. And a later version focused more on tasks that were verified by humans to be unambiguous. So this kind of reduced the number of tests that were being tested. And performance here increased dramatically to 70%, which may partially be due to the removal of the harder ambiguous tasks, but also to improvements to the models themselves and the scaffolding as these frontier models are constantly being updated. And one potential issue to consider with this type of benchmark, and it's important to consider with benchmarking in general, is the potential for data contamination. Since these models are exposed to large amounts of data throughout training, it is possible that the benchmark was exposed to some version of these repositories during training, which makes the benchmark actually less reliable as a true test of performance on truly novel tasks. And a later version of SWE-bench Pro utilized proprietary code bases in order to try and kind of mitigate this issue. And this showed decreased performance, which does suggest that contamination may have been a partial issue in the initial evaluation. Next, I will talk about LabBench, which was designed to measure various capabilities specific to biology research. And it consists of 2,400 MCQ questions constructed with a combination of LLM prompting as well as question generation and curation by scientists. And it included a variety of tasks. So for example, it included some questions which involved showing the model a figure or a table from a publication without any associated text and asking it questions that require interpretation of particular features of the figure or the table. It also included questions that require search over a biology database or over supplementary material in a paper, as well as questions that require reasoning over molecular sequences or retrieval of information from scientific literature. And for this information retrieval, there was a focus on esoteric findings in recent papers in order to try and avoid the potential of the model just answering from memory or simple retrieval from the abstract. And finally, it included questions that require reasoning over scientific protocols. So these were protocols that were actually manipulated by either modifying or omitting a step. And the model was asked questions related to generating hypothetical outcomes of these modified protocols, as well as identifying the fix required in order to generate the intended output. And there was also some questions related to complex real-world cloning scenarios. And performance in the initial evaluation varied across tasks, but generally it was below human performance in most cases. But again, an important note to keep in mind here is that this evaluation included looking at frontier models without any tool use augmentations, which establishes a lower bound for the potential performance, but it is possible that properly scaffolded tool augmented agents may achieve better performance. And again, as I mentioned, frontier models are always being updated, and so performance may vary with time. So there are a few limitations that the authors actually mention in the paper where they introduced this benchmark, and I think they're important to consider. The first is a limitation of using MCQ benchmarks in general, which is that the model may be better at eliminating unlikely answers than actually identifying the correct answers. So essentially, it's using good test-taking abilities in order to compensate for poor reasoning abilities. And for this reason, it becomes very important to design high-quality distractors with these questions, and also to incorporate open response evaluations, which are difficult to scale, but are really important for evaluating agent reasoning. The second is what I just mentioned, which is that the models may be sensitive to prompting and scaffolding, and so performance may vary based on the system setup. I think we have a question. Yeah.
A simple question: was this research done on just the LLM, or actually the agent with a loop and all the scaffolding?
Yeah, so this particular evaluation was done on just the LLM. So they did not augment any, with any tool use, which is why it sets a lower bound for the potential performance.
Loop, right? So it's just one shot.
I believe it was just one shot, but I'm not entirely sure. Yeah, and then another limitation is that the release of this benchmark may again produce the risk for contamination in future model evaluations, because newer models may be trained on data that includes either this benchmark or descriptions of this benchmark. For this reason, they keep 20% of the questions actually private, which may help with detecting contamination in future evaluations. And finally, while this benchmark evaluates core competencies for biomedical applications, still biomedical workflows include the integration of many of these types of tasks, which is not being evaluated here. So just to sum up,
can I ask another question on that?
Yes.
Can you go back to the performance figure, please? Yeah. So did they go into, what were the types of questions it messed up on? Like big QA looks like pretty bad in general. Like, why is it having a harder time there versus, say, the table? Like, and then did they also ask the model to give like a how confident are you in your response?
I don't think there was a confidence measure in terms of why particular tasks were potentially performing worse. It's possible that something like, for example, the figure, which requires kind of multimodal reasoning, becomes more difficult without any tool augmentations, for example. And then One more thing to know actually is that if you look at the accuracy versus precision, the precision figure specifically includes, the denominator is questions that the model agreed to answer. So they would always give the model the option to just opt out if it cannot answer. And that's why you kind of see the difference between these two. And there was also a difference in kind of, the types of questions that the model would simply think, it's too complex to be answered. Yeah, so just a quick summary here is that initial evaluations revealed low performance for frontier models, but that has increased since publication. And there was also the development of LabBench 2, which extends the difficulty of some of these tasks and also includes some open response questions to try and get at some of the limitations of the initial benchmark. And then next, I want to discuss bioinformatics bench, which consists of 61 real-world analytical scenarios. They refer to these as capsules, which where the agent would kind of be required to execute multi-step analysis and information interpretation in order to answer a particular set of questions for each scenario. And these were developed by scientists that were asked to either reproduce findings from publications or to perform de novo analysis on data that they provide. And each capsule would consist of a hypothesis or a research question, a set of input data, analysis code written by the scientists who created these capsules, the result, as well as an answer to the question of whether the hypothesis is supported by the data and the analysis. And the final set of curated capsules spanned various subfields in computational biology. And then they used a Claude model to generate an initial draft of multiple choice questions related to the analysis in each capsules. These were then reviewed by analysts who could approve, modify, or reject questions in order to create a final list. And they were particularly designed such that the questions could not be answered based on all agent knowledge alone, but rather would require the agent to interpret the research question, perform data exploration and multi-step analyses, and interpret the result in order to be able to provide an answer to those questions. And then agents were asked to perform the required analyses in each capsule in order to answer the related questions. And they were evaluated in two different settings. The first is an open response setting where the agent answers along with ground truth were given to a separate language model that was then asked to generate a binary score. And there was also a multiple choice setting where the analysis that was produced by the agent, its open responses were again given to a separate LM along with the questions and multiple choice options. And then this second LM was asked to select the answer based on the analysis and the open response answers from the original agent. And in both of these cases, they performed 5 parallel runs in order to account for potential stochasticity. And again, they observed that performance was generally low. And the baseline here actually was set by asking an agent to just answer the questions based on pure reasoning without performing any analyses. And one thing you can see here is, for example, in the middle with the MCQ questions with a refusal option, essentially the agent can select this option that says there isn't sufficient data. This shows near random performance, suggesting that the models have a tendency to opt out of complex analysis when they are given that option. So even though this benchmark is an attempt at measuring agent performance in the integrated tasks required for a full bioinformatics pipeline, it still has several limitations, including a small sample size of 61 capsules. And also the use of LLM as a judge is itself a step that may introduce some reliability concern and might require some amount of benchmarking itself. It is still an example of the evaluation of result rather than procedure. So it is still possible that the analysis the agent produced actually has some issues that were just not kind of represented in the final answer. And finally, so one strength of this benchmark is that it includes multi-step analysis workflows, but that also means that it incurs additional computational costs, which makes it more difficult to test a large amount of models with this kind of benchmark. So that's a trade-off that, again, is important to consider. And so to wrap up, I want to kind of give a quick summary of the capabilities and potential failure mechanisms of these systems in biomedical applications, and then kind of give a description of specific points that I think are important to keep in mind when developing these systems. So as I mentioned in the beginning, I'm sure you have seen many papers that demonstrate agent abilities across different tasks in biomedical settings, such as literature search and database queries, data analysis, the generation and interpretation of hypotheses, as well as experimental design. And also a growing number of papers have shown that, agentic systems may orchestrate multiple multi-step research workflows that involve the integration of many of these types of tasks. However, as we've been discussing, these agentic systems are actually very difficult to benchmark, especially when you think about real-world deployments. The first important issue that we should consider is contamination. So the agent may have seen the benchmark being used or some closely related tasks during training, which makes the evaluation procedure itself unreliable. And so it's important to take steps to mitigate this issue, for example, by testing on tasks that are resistant to memorization alone, and also by using benchmarks that are based on previously unseen data, kind of analogous to how you would always want to have a held out test set in any machine learning model that you develop. There's also the issue of reliability that we just mentioned, where an agent may give different answers on different runs. This could be a result of model stochasticity, but also just differences in prompting and scaffolding. And so it's important to evaluate the agent over parallel runs. And for example, the Tau bench paper introduced the pass-hat-k metric to try and kind of evaluate reliability over multiple runs. The third is, as we've been discussing, it's important to measure the process and not just the final result, because, especially when we try to apply these models into novel cases and we don't know what the desired result is, it's important to make sure that the workflow itself is sound and does not lead to policy violating steps. And then finally, if you want to develop multi-agent systems, which can be helpful in the biomedical setting, then evaluation becomes even more difficult because there are multiple components that need to be tested. And we also need to measure additional capabilities, such as the coordination and communication between subagents. And there are also several failure mechanisms that are important to keep in mind. The first is that hallucination is still an issue, even though this has improved with more recent models. It's still a critical issue, especially in the biomedical setting where the stakes can be high. And it becomes a bigger problem in long workflows where some initial hallucination might actually propagate through the workflow before being detected. And the second is that reasoning traces may not actually reflect the procedure used by the agent. And so it makes it difficult to have proper oversight of these agents. And given that safety and security violations are possible, it is important to potentially have hard-coded steps in order to try and prevent such violations, such as, for example, enforcing kind of checkpoints, to approve actions, or working in a sandbox environment as well, where you prevent the agent from potentially overriding files or deleting data. And then also self-reflection without external feedback may itself not be effective. And so this again creates the need to have checkpoints for human feedback. And it also means that when you have long-term workflows, it is possible that premature commitment to planning may not be properly corrected and may lead to cascading errors through the workflow. And finally, a consequence of all of this is that benchmark performance does not necessarily reflect the deployment performance. And so that's very important to keep in mind when actually developing systems for potential deployment. And finally, just a few steps to keep in mind at at each step of the procedure of developing these systems. First, in terms of problem definition and setup, it's really important to define the task precisely and to define measurable success criteria. And this will be important for system design and evaluation procedures later on. It's also important to identify what input data you want to make available to the system and think about any potential, for example, de-identification that you will need to do in order to protect private information. And also to think about what private benchmarking procedures you might want to use and whether that would require potentially leaving some portion of your data as an unseen set before running the system. You also want to think about what failure modes you most want to avoid for your application so that you can take steps to either mitigate or detect those. And you want to identify specific points for human intervention and oversight throughout the loop. And then when it comes to the system design step, you want to carefully design the system prompt in order to define the problem and also define what are your desired outcomes and potentially output modalities, whether that's figures, tables, or text, and any rules that you want the agent to follow, for example, in terms of reasoning or verification, and potentially providing few-shot examples of the behavior that you want. It's also important to design the scaffolding very carefully. As I mentioned, this is essentially what manages the entire loop. And here is where you can enforce particular types of reasoning or verification, as well as also manage the agent memory. And you can also include hard-coded safety mechanisms at this point, includes checkpoints for human oversights or approval gates, and can also include write restrictions for files and databases and backing up files prior to running the system. This step, you would also create tool schemas. And again, it becomes important to very clearly define the use for every type of tool that you want to make available and what are the required arguments so that the agent would be more likely to call these tools correctly. And if you're using a multi-agent system, then defining sub-agent roles very carefully and also defining rules for how the orchestrator would kind of manage those sub-agents. And finally, for the monitoring and evaluation step, keep in mind that reasoning traces, while helpful, are not necessarily a faithful explanation of what the model did. And so it's important to have these hard-coded safety mechanisms that we mentioned earlier. And it's also important to try and evaluate the process quality and not just the outcome. So looking over, for example, the code that the agent wrote, and also evaluating tool use decisions and verification step effectiveness, as well as the safety of the procedures that are being used. When possible, to perform parallel runs and report kind of a pass hack K metric, for example. This can also be important, especially if you're trying to develop a system for deployment. And finally, having some sort of private or time-restricted benchmarks that allows you to benchmark the agent performance without the risk of contamination. It can be quite important. So this is all I had. Thank you all very much for listening, and I'm happy to take questions.
Yeah, if anyone has any questions, I'll just pass around the microphone to people.
Is there a benchmark for multi-agent systems, or is it too new?
A benchmark specific, I'm sure that people have, developed specific benchmarks that looks at these. I mean, in general, you would want to kind of evaluate how the coordination and the communication between them works, for example, and how specific, whether they really stick to like the particular tasks that you try to assign to each of them. And it also becomes important to then try to look at how the orchestrator kind of divides the subtasks. I don't know of any specific benchmarks off the top of my head, but there's constantly new papers in the field, so I'm sure there's something.
Is that working? Okay, great. So I just want to say, like, I do work in part at Future House, so like I know about some of this stuff. And some of the conversations we had there, which I'm curious about your opinion on, and maybe later after the next talk as well. But how much of their emote is this at this point compared to Claude Code or Cursor or any of these things? Like in terms of building up these pipelines within those versus building up independent ones. By the time that you build this or build an agentic system for biology, how far behind is just the latest release of Mythos or something like this?
Yeah, that's a great question. I mean, I think there are, so to clarify, the question was like, essentially by the time you build the system, how far behind are you from like the current frontier model, essentially. Given how fast it's moving, yeah, you could be quite far behind. And maybe it actually becomes important for you to then utilize some of these models in order to help you kind of speed up development, while again, keeping in mind that oversight is important. And so, it can help you speed up the development of your system, but also you want to make sure to always review the code and like the specific safety mechanisms that you want to have. But I feel like in this field, you're almost always going to be behind something because there's always something new coming up.
I'd like to rephrase that question a little bit. It's not like who is behind who, but it's like, is there anything special on your biomedical application side, or actually the cloud code can do everything that you are doing? So I think that's two different questions. It's like, I think you must learn a lot while trying to build a system specialized in this bioinformatics, biomedical tasks, right? And those are probably often not cared by those general foundation model. Like talk to ChatGPT about the bio question might be different in talking to your specialized or trained agent with certain skills. So I find this direction might be more interesting, but I'm curious about your thoughts.
Yeah. So specifically, you're saying while the models are basically being improved all the time, they are not really being improved with biomedical applications in mind, and so they might still be behind in those.
Yeah, but do you also find, there's something special while building this specific in this domain that cannot be easily like just replaced or like just overwritten by the major popular foundation models or agents?
I see. Yeah. I mean, I think, again, you're right. It does become an issue when you're talking about this very specific use case because they aren't necessarily being developed with this in mind. And so it is very important to actually utilize your own domain knowledge in order to make sure it is doing the correct things. And also in thinking about, for example, what I mentioned briefly in terms of skills, like actually telling the model that this is the correct way, for example, to perform quality control for this type of data that I'm using, because this may not necessarily kind of be available in terms of native performance. And so I do think that, relying entirely on just the improvements of these frontier models, it's not sufficient. You definitely have to kind of utilize your own domain knowledge. And these models give you improved reasoning abilities and potentially improved answers, but incorporating your own knowledge is really important, and overseeing what it's doing is
really important. We have a question from Zoom from Krista Owen. For biomedical large language models with reasoning capabilities, how to evaluate reasoning traces to ensure correctness. Wrong reasoning could still lead to correct answer output. I think it's something you mentioned.
Yeah, exactly. Yeah, and this is the issue where reasoning traces are not necessarily sufficient for oversight, and so again, there is still an important need for human intervention and review of their specific procedures rather than just the answer itself. Because exactly that, right? Like wrong reasoning might still lead the model to have a correct answer, and the reasoning that's being shown may not necessarily reflect everything that the model actually did in order to provide that answer. And this becomes an issue when you try to apply it into novel tasks where you don't necessarily know where the answer is. And so looking through the reasoning traces, specifically with your own domain knowledge in mind, becomes important for evaluating whether it's potentially doing what you actually want it to do or not.
Great. So if there's one more question.
A question about the evaluations and kind of the community and whether there are are there any central organizations that are starting to create kind of repeated and relatively unbiased evaluations of these types of systems, in the sense of the CASP, right, was there for decades in protein folding? Is there any thought or kind of evidence that there's something emerging in this space rather than kind of lots of point benchmarks and evaluations?
I'm not sure personally if there's something like that happening. I know, for example, Future House, for example, tries to kind of produce very different important benchmarks for this setting, specifically with a focus on the scientific workflows. But I'm not sure about exactly a central organization that you're describing. Potentially there is, but I don't know.
thanks for the nice talk. I have a question about like just again like how agents will be used in general. And so I think these are sort of marketed as like now I have these expanded capabilities that I didn't have before. So I work in omics, but now I can go and design a small molecule that binds whatever protein. But that is probably still a bad idea. But now I can go and make that molecule and put it in my methods and have the code. And so how do we guard against like essentially non-experts doing things that actually require expertise and it going into the literature and kind of flooding it in a way?
Yeah.
Because that also will have repercussions on the models now, right? So like how, so again, it goes back to my original question is like, if I have a methods and I'm publishing, you know, my, whatever my agent did, and I say, I looked at the code, but what if I'm like not a person who works in protein? So there are things that are just obviously I'm going to miss. So how do we as scientists think about that problem? And like, what are things we can do?
Yeah, I mean, I think that's a very important concern. I think in general, again, I personally don't believe we should just kind of entirely rely on these models, and we should still have experts. So, for example, in the setting you're describing, where you're going into a field that is not actually your own field, still reaching out to someone who is actually an expert in this field to help you overseeing what the model is doing, even if you used it to actually generate the analysis, is going to be really important. Potentially, the use of these models would help you speed up your move into this field because, you're going to that expert with something in hand that you can talk about and review. Essentially, it might help kind of speed up the workflow and be able to perform these projects potentially with less scientists actually kind of doing hands-on work. But I, again, think like it's really important to, one, continue to have oversight across the board, across the whole workflow. And also, of course, like, with publications being very transparent about what is actually AI generated and what is, was actually performed by scientists. Thank you all very much.
Our summer speaker today is Shreya Johri. Shreya completed her PhD in the Biological and Biomedical Sciences program at Harvard. She is currently a postdoc in Eli Van Allen's lab and has received the McDonough AI Fellowship at Dana-Farber Cancer Institute. Today, she will speak to us on evaluating AI agents for biological discovery. And with that, I'll give it over to Shreya. Thank you so much for coming.
Thank you so much for having me here today, and excited to talk about our ongoing work on evaluating the autonomous and copilot limitations of AI agents for biology. And as we all saw in the primer talk, there have been so many papers that have been coming out, and there's a growing number of literature that on agentic AI papers for biology. And so it becomes, it's following trend in other fields too, and it becomes important to know how to evaluate these models, as Maha really laid the primer for. But evaluation in biology is also hard. There are multiple aspects, multiple angles in which we need to think about evaluations. One of the first things that comes to mind is, can AI agents perform complex analyses reliably? And I want to stress on the word complex because this word is actually deeper than just like the word itself. It can be multimodal analysis, it can be multi-step analysis, and in many different ways, a complex data set. So are AI agents actually reliable if allowed to do these analyses? Another question that comes to mind is, can AI agents answer targeted biological questions in a robust manner? By robust, I mean if the agent finds something in one data set, is that also possible to verify in a different modality or a different data set of the same tissue type, same cancer type, same broader morphology? And finally, how do AI agents integrate into computational biology workflows, which is something that we've all been thinking about. And so I just want to start first by talking about computational biology workflows. So these workflows are inherently multi-step and they're iterative. So let's take an example of a single-cell RNA-seq dataset, which means that every cell of the sample was sequenced for the RNA. And so we have a cells cross genes matrix. Typically in the workflow, there'll be a quality control step, there'll be a somewhat standardized single-cell RNA-seq pipeline that would be run. people would then do cell type annotations. But one thing that a lot of the benchmarks actually miss is the iterative part of this whole step. No one who's a computational biologist one-shots the analysis. You typically have to repeat the steps multiple times. As an example of why you would need to do this, for example, you did the first pass, you reached the cell type annotation phase, and you realize that some of the cells actually did not get filtered in the quality control step, they escaped. And now they're making your analysis less, they're interfering with your analysis. So you would ideally want to go back, remove those cells because they are low quality cells or whatever they are, or maybe they are doublets, and then you want to repeat the pipeline again. So this iterative aspect of computational biology is very important to monitor. It's very important to actually observe in AI agents and how they behave and if they behave like this. Another aspect of computational biology is the multimodality. So whenever we are analyzing a data set, we don't just look at printing tables, or we always make plots. And plots are a very important signal for us to identify outliers, to identify samples that may be behaving differently. And so in order to assess models comprehensively, it's important to actually also provide these, have a loop that models are able to look at these images to, and have that multimodal aspect embedded in the system. And then finally, multimodal integration. So multi-omic integration. So multi-omic integration always gives more information about the samples that you have. And I can give an example here. For example, let's say that you have only the single-cell RNA-seq data set. So the RNA tells you about what genes are expressed, but it does not really tell you anything about the regulatory regions. So it's possible that there are, what looks as one cell type or one cell state is actually two distinct cell states which have different regulatory behaviors. One of them might have open chromatin for certain kinds of transcription factors, which would be affecting the way some genes are expressed, but overall the expression is still looking the same. These kind of nuances actually become really important when thinking about questions like therapy resistance or very targeted biological questions. And so definitely looking at how AI agents are able to do in multi-omic settings is also important. So current benchmarks overall, we believe, are limited in formulating the real world computational biologist's workflow and understanding how these models fit in. Where are humans in this process? Are these models reliable when left alone? Are they adding any value when humans get involved to the human who has worked on this data set before, is new to the working on this, and so on. So for this kind of evaluation, we define something called an M3A evaluation framework, which stands for multi-step, multimodal, multi-omic agentic framework. So at the core of it, it is an LLM, which is initialized with the Opus 4.6 model. This was released in February of this year. And in addition to this we have a scaffolding around it. So the AI agent is deployed in a standardized execution environment so that the results are always starting from the same point. It has access to specialized tooling. Specifically, in our case, we were interested in looking for single-cell multiomics aspects of how these agents are able to perform. And so single-cell RNA-seq tools, single-cell ATAC tools, web search tools, coding tools. The M3A evaluation framework has very strong multimodal context integration. So any images that are generated, any text that is outputted from the tools as a print statement or as output is also backfed. We maintain persistent data state, which is actually also important in making sure as you progress through the analysis, if you've done some transformations, that gets maintained. And then finally, we also want to understand a little bit about how these models are making decisions. While this process is obviously post hoc and we're not looking at the model weights, it gives us some insights into how these models are making decisions. So we try to log why the models are calling different tools or why they're trying to write different kinds of codes for the analysis. Let me walk you through an example of how this is set up. So let's say that the agent has been given a task description and it is deployed in a standardized execution environment, which is basically powered by a Docker container and has access to this data set. So it starts with some exploration of the data set. It's able to do this because it has the ability to code and it has that Python coding tooling integrated. Then after exploration, the agent decides to do some single-cell RNA-seq-specific analysis, and in the process, some of the ways in which the model does the analysis actually modifies the dataset. So this dataset then persists, and there's also some extra assets that might be created. There might be tables, there might be figures, and so on. So all of this is different data assets get created in different steps as the agent is doing RNA analyses. Maybe it goes on to do data analysis, and more data assets get generated. And once it has done maybe some of the data exploration that it has, then it might also go out to look at maybe related papers regarding some of the tasks that the agent was doing. It might also search the web, not just necessarily PubMed or bioRxiv, for some general therapy-related information, some general information that's not necessarily appropriate for finding in a paper. And after all of these steps, the agent then finally gives a final response that contains it hopefully trying to solve at least partially the task that was given to the agent. So you will notice that the agent was using different kind of specialized tools at different points in time. It's possible also that the agent, despite having access to the single-cell RNA-seq tools, decides to write its own code. And that is a part of agent behavior that we have seen happening. You would notice that the persistent data state also gets maintained. So anytime any modification is being done to the data, either that's stored as a layer of the A-data object, or it's stored in a way that if the agent wants to backtrack, it can actually go back a couple of steps and then redo from the starting point. There's managed multi-step multimodal context integration, which means that all the tools, their parameters that were used, any outputs generated from the tools, any images that were generated, any intermediate reasoning text that was generated is all maintained as one long conversation as the agent is reasoning. And then finally, the step-level telemetry actually happens at the tool call level step. So right after the agent has made the decision that it wants to use this tool, but before that tool gets executed, so the agent hasn't really seen the output from that tool and hasn't really executed that tool, we log three different things related to that decision-making process. So why did the agent choose this tool? Did it consider any alternative tools, or did it consider writing its own code instead of using the tool? And then finally, what the agent expects would come out of using this tool. So using this M3A framework, we do comprehensive benchmarking on single-cell multiome samples spanning 15 different cancer types and over 240 patients. We evaluate on different kinds of tasks. So these tasks span clinical, pathology, molecular, and I'm going to talk about each of these in detail in a bit. When it comes to, there's one that's the task description. When it comes to evaluation, we also take a multifaceted approach in trying to understand when these models what these models did right and what they did not do right. So it's a combination of human evaluations with computational biology experts and oncology experts, and also multimodal evaluations in a different data set. So in this case, we use TTGA. And then finally, we try to understand the ways in which humans interact with these models and how that influences how these models are able to reason. And I'm going to talk about each of these specific interaction modes in a bit as well. So let's come to the first question of, can AI agents perform complex analyses reliably? And I'm talking here about the single-cell multiome pipeline, or the generally executing pipeline aspect of the number of things that we do in computational biology. So for this specific task, we used on the y-axis, you are seeing the different cancer types that were part of this evaluation setup, and they correspond. And the x-axis is the number of patient samples per cancer type, and we had a total of 207 patient samples here. For each of these patient samples, we had a single-cell RNA-seq matrix, we had a single-cell ATAC-seq matrix, and authored cell type annotations, because all of these samples had been taken from a previously published work. This work was part of the HTAN consortium, and one of the reasons why we picked this is because they had actually standardized to some extent the way the samples were generated, as well as somewhat annotation, the way these were annotated. So we could actually compare across different cancer types, not just within that specific cancer type. And we wanted to have that cross-cancer comparison. So what really happens in this is the agent is provided with the single-cell RNA and single-cell ATAC matrix. It is provided with, it's described a task, which is identify the major cell types present in the data set. And you will notice that the task is deliberately kept. This is not the exact task. This is the exact problem statement, but we also provide, in addition to this, we tell the agent that there are these data objects that are present in the environment. So it knows how to look for them, because I can't really provide the agent in anything else. I can just point it to that this is the data object that's present, but we deliberately do not make the task prescriptive. Like, I do not tell the agent you have to do A, B, C, D steps to actually do the task, because we want to see how agents are able, what agents are able to do on their own. And when they do it on their own, how matched is it to the standard way in which we do things. After the agent does the task, it produces three different things. One is the code that it used to do all the analysis. So oftentimes it produces a tool. So that tool is specifically, it's basically a Jupyter notebook. So the tool call is referenced there. If the agent decides to write its code, which could be that the agent is exploring the data set, or it decides to do something that a tool is not available for, it will write that code and we save that code. Step level reasoning information is available. And then finally, the task leads to the cell type annotations that we want to check. And the way we validate all of these is we do a manual review of the analysis code, the step level reasoning, and for the cell type annotations, we actually compare to the author cell type annotation that we have from the publication. So one interesting thing that jumps out is that agents iteratively actually refine cluster annotations. So on the x-axis here is the number of, on the y-axis here is the number of scrutiny steps. And I'm showing this just for one data set right now, which is melanoma. And you would, each dot here represents one of the clusters. And each of these, I have labeled broad, I've colored this based on broader cell types, but each of these clusters has like been assigned a more specific annotation by the agent. So you would notice that some of the clusters at the bottom, the agent actually decided pretty early on that this cluster was, in this case, immune. But then for this cluster, the agent actually spent 11 steps of iteration in order to identify it as a stroma cluster. So an example of how this looks like, so this cluster at the top that I've labeled as stroma is actually cluster number 14. So the agent at this point has done the broad cell type, like dot plots to see broad expression of different marker genes. And then it looks at specifically some of the, it comes up with, what I want you to note here is that it comes up with a hypothesis that the top PEG genes suggest dedifferentiated melanoma or neural crest. It does some analysis, then in the next step it says this could still be mesenchymal or dedifferentiated melanoma, but it has these X genes, so it could also be melanocyte. Then it checks for those melanocytic specific markers, and it finds that those are actually pretty low. And so it says, this doesn't look like melanoma, and let me check if it's a different cell type. So the agent is actually having the capability to after it has made a hypothesis that this is probably this cell type, it is actually also being able to backtrack and agree that this is probably not that, and it is open to exploring other hypotheses. And then the agent then goes on, looks at other different marker genes, and goes on for like multiple steps. But I just wanted to point that this is like iteratively happening for different clusters in different ways. And this happens across all cancer types. It was not just like a melanoma case. You will notice that different kinds of cell types, the top ones are always annotated by the name. They always have taken a lot of time for the agent to decide what it is. But all of these are different. It's not like there's one cell type that the agent has been having difficulty in doing. And when you look, just look at the overall accuracy, not just like, this was scrutiny, which was more of the, how did it get there? But when you look at the overall accuracy, which in this case, we are using AURC, you see that is very high across most of the cancers, which had a good number of samples. It's between, for the top few different cancer types, is between 0.9 to 0.99, which is pretty high. But when you actually look at a better metric to account for rare cell types that could have been present in the sample, which is done using a macro AUC, you see that the accuracy actually, like metric actually drops a lot. What this means is that overall, the agent was doing a fine job identifying cells. But those cells, but it was misannotating a lot of the rarer subtypes in that tumor sample to a different cell type, which was incorrect. And so I guess the takeaway is that while the agent was good at broader annotating cell types that were present in more abundance, rarer cell types were often mislabeled. And some other observations from generally like having looked through the manual review of how the code was written, how everything was done. So we find that there was no iteration on the analysis of the pipeline step. There was only iteration on, is this cell type or is this not the cell type? Like it would never, once it has done the clustering, it would never go back and redo maybe the resolution of clustering. It would never redo any of the quality control or anything. There was no web search or literature review tools that were used for selecting the gene markers either. So the agent was only relying, every time it was iterating, and you will notice that it iterated for sometimes like 12 steps. It was only using its own domain knowledge to check if that marker gene expression, which correlates with some specific cell type, is present or not. And step-level telemetry, when we look at those, they also do not indicate that the agent was considering using web search at any point in time. So the agent believed that I had all the knowledge that I need to do this task. And then finally, the single-cell RNA part of it was the standardized. It's pretty standard in how people do it. And we were very curious to know what the agent was doing with the single-cell ATAC data, because it actually gives very important insights into the cell states. And so we found that from the agent was using that data for independent clustering only 4 of the 10 times for the 10 different cancer types we had. And in the rest of the cases, the agent was just using it to spot check. Like it would check that, it would find that this is the specific cell type. It would go back to the single-cell ATAC, single-cell ATAC data to check for some specific transcription factors and then be okay. But it wouldn't independently look if the single-cell ATAC matched with the single-cell RNA specific. So it seems like the agents, they are doing some things, they're not doing some things. And it's, again, being vigilant about what we are asking them to do. But the next thing that we wanted to ask about was, for targeted hypothesis exploration, are these AI agents producing robust responses? Which is the step after you've run the pipeline, when you actually have like maybe a hunch that this could be happening, and then you go down, look at multiple different data sets that would give you a signal, or you would consult someone who has specialization in that aspect, and so on. For this, we again use the HTAN dataset. In this case, we removed the CCRCC and GBM cohorts because they just had very few number of samples, and we wanted it to be more larger number of patients when we did this. And as before, the single-cell RNA, single-cell ATAC, or the cell type annotations were present. But in order to phrase that task as a very targeted question, we ran CNMF on the single-cell RNA matrix. CNMF is a method which is similar to non-negative matrix factorization, but it gives you, at the end of it, gene programs. And each gene program one cell can express multiple gene programs, so it's not that they're necessarily orthogonal. But these gene programs represent independent, dominant programs in that specific sample. So each of these gene programs has different types and different kinds of genes present. So after we have all of this set up, we provide these 4 different inputs to the agent. And the task is, again, kept very minimal. We say that predict the CNMF program with, I'm giving an example in this case from the breast cancer cohort, and one of the tasks that we used to explain better what we exactly did. So the task is defined as predict the CNMF program with relevance to ER-driven luminal identity in breast cancer. ER-driven luminal identity is a very strong and very well-known biological program. So we expected that the agent we expected to find this in any CNMF programs that have been generated in an unsupervised fashion. And this was our design strategy for all the evaluation, all the targeted questions that we had for each of the cancer types. So the agent again gives analysis code, step-level reasoning, and the ranked CNMF programs, which basically is just like a ranking, that CNMF3 is the topmost program that was found to be most associated with ER-driven luminal identity, then CNMF1, and so on. So how do we then validate whether this, like, there's no ground truth here, because it's a new problem, it's a new question that you've been interested in. So one of the ways that we thought we could do this validation is by testing it in a different data set. So we took bulk RNA-seq samples from TCGA of patients who had been diagnosed with breast cancer, and we used the CNMF programs to score these bulk RNA-seq samples, which using SSGSEA. So SSGSEA is a very popular method that is used for scoring bulk RNA-seq. Some of you might already be familiar with it, but it involves ranking the genes based on expression and using that rank information to generate a SSGSEA curve. And the area under the curve then corresponds to the GSEA score. And that score is how we are, so basically for each, after the scoring, we have a matrix of patients' cost programs, and we use the SSGACA score of each of these, the values of these like matrix elements are actually the SSGACA scores. After that, we extract the ER positive status from the IHC data that is associated with TCGA. So this is a ground truth. Someone has already done IHC on the same TCGA bulk RNA-seq sample. So we have a very solid way to verify that. And we calculate the AUROC per CNMF program for ER positive versus ER negative samples. So the AUROC then is used as a way to rank the CNMF programs in the order in which, like, the program with the highest AURC has the most discriminative power between ER positive and ER negative patients, and so it's ranked the highest, and so on. So when we compare what the agent came up with and what the evaluation TCGA yielded, we find that both of them actually did identify the same CNMF program as the top program that is most associated with ER positive luminal identity. But this was actually a somewhat easier task, and we expected the model to be able to identify this. So this was more of a positive control. We defined many different tasks in this way, in which we are able to verify the target in a very robust fashion. So like we used ER positive status from IHC, we also had other data associated with these TCGA samples, for example, overall survival, which is a clinical task. We could also look at histological grade, which has been annotated by the pathologist in the pathology report, so that could be extracted from those reports. And so we basically took all of the information from a very verified target in our validation cohort. So we find that if you just look at the top CNMF programs that were identified across these different tasks, the AI agent was performing, the dotted line here represents the random baseline. So if the agent was randomly guessing any program as the top program, that would have like 0.11 accuracy. But we find that the agent is definitely performing better than the random, but it's still very far away from actually being very useful. There is definitely signal. So we see 0.23 overall accuracy. And when you break it down by the different kinds of tasks that we tried, we see that the clinical tasks, which were primarily based on overall survival, are actually most easiest for the agent to identify the top CNMF programs for, followed by pathology, and then followed by molecular. I wanted to show a few examples exactly of what the agent, what some of the tasks that we had, what the agent identified, and where these programs are actually ranked. So let's take an example of one of the clinical tasks. So this is head and neck cancer, and we have the overall survival task that we tried. So one here indicates that the agent actually identified the CNMF program, which was actually also the top CNMF program that was identified in the validation cohort. if you look at the PDAC cohort, which is the pancreatic cancer, we see that the CNMF program that was actually supposed to be the first was actually ranked by the agent as number 3. So the agent misses the actual ranking. And you can similarly look at other tasks in pathology, for example, lymphovascular invasion, resection margin status. We looked at different histological grades, different kind of subtype differences also. And for some of them, it was surprising that the agent was not able to identify a very clear signal, because some of these subtypes are they have a very distinct biology. But for some of the molecular tasks, we were actually surprised that the agent got it right. For example, for melanoma, we found that when we specifically asked the agent to identify a program, the CNMF program that was most associated with BRAF proliferating mutations, it actually identified it correctly. It did the same for NRAS mutant subtype. And then similarly, it was a little bit off for the UCEC cohort. And there are multiple examples of also for the things that it was not right about. So this plot is showing not just the top one, but the overall ranking of the agent. So because we have the ranks from the actual ground truth, which is from the TCGA, we have the ranks from the agents. We're able to actually calculate a Spearman correlation that can tell us how much these ranks match with each other overall, not just the top one. And we find that overall CNMF program concordance is actually highly variable between cancer types. And if you look at different kinds, like specifically for different task categories, like molecular, pathology, clinical, they're still very variable. In some cases, you do see some signal. In other cases, when you average out across different tasks that we had, that signal kind of becomes very negligible. So agents are able to do some of the things, but agents are also not able to do some of the things. And it's still, when we look at the reasoning, we see them making very reasonable assumptions and writing very reasonable code that would have been written if a human was doing it. But somehow in the middle of that reasoning, the agents somehow go down one track or chain of thought, and that sometimes leads to them probably not coming to the right conclusion. I want to caveat this by saying that it's also possible that some of these are, because it's a different data set, they might not actually be, the biology might be slightly different, but we specifically chose tasks that were supposed to be very prominent biological axes. And so that should be at a minimum in our opinion. When we look at specifically, because for each CNMF program, we can actually identify what that CNMF program stands for. So we wanted to see if the agents are specifically favoring some kind of programs as always projecting up, versus if there are some programs that the agents are always discounting regardless of what task they're doing. So we calculated the rank difference between what the agent ranks higher versus lower for different kinds of CNMF programs across the cohort. So I've grouped it into broader biological axes here, but the programs are much more specific when you actually look at the annotations from the agent. So we find that the agent will often rank the stress so if you go more left, the agent is over ranking, and if you go more right, the agent is actually under ranking that program. So for example, for the stress or translation or the EMT or stroma program, the agent is often ranking it higher than it was in actually the ranking, because it thinks that these programs are maybe more often associated with with whatever task it was assigned. And I think kind of comes to the general, I guess, general kind of inclination that EMT mostly is always associated with something specifically aggressive in cancers.
I have a question here. For these kinds of gene sets, there are sort of, I guess, like known issues with the way that these are constructed that have to do with things like, is there a lot of overlap between gene sets? Is there, like, how much, like, how are the gene sets themselves validated? And I was wondering how you might have approached this here, because when I see this saying EMT, frankly, I see a lot of humans, just saying, "Oh, EMT shows up. I did GSEA, EMT, it must be EMT." When does the biology make sense? How do we do that? The EMT signal in Hallmark, for example, is very I don't know. I've come to become skeptical when I see it somewhere. What does it actually mean? So I was curious if you had thought about or benchmarked somehow how good you expect any of these particular gene sets to be for a narrow scope in the first place.
That's a great question. So all of the gene sets actually have not been derived from any of the existing databases because of the problems that you described. The gene sets have been derived from the single cell RNA-seq cohort that we had, and that derivation has been done in an unbiased fashion. So the non-negative matrix factorization actually identifies, it factorizes the gene matrix into two components such that all of the elements are non-negative. And that then you can map. So essentially you get CNMF, which has a set of genes. Then you run GACA, the models have run GACA on it and identify some top GACA, depending on what set was used, some either based on Reactome or KEGG or MSigDB. And then based on the existing programs that have been annotated, it assigns a value to the CNMF program. So it's possible that some of the things that have been defined in literature as EMT have overwhelmingly shown up in a program, and that's why it was annotated as EMT. And so we are just going off of what the literature or the database that was used to annotate that. And then I've clubbed it as broader category. The agents described it more specifically in most of the cases, not necessarily EMT. But I'm happy to show you some specific examples if that helps. But I agree that there's always that problem of the gene sets could be too broad. That could also in a way affect the CNMF that you are doing.
We also have a relay question on Zoom from Jeffrey Sowell. How biased are the AI rankings of CNNF programs by the signal intensity of the program, number of genes in the program, etc.? i.e. programs that have a high expression may be more identifiable than others.
Yeah, that's correct. And that's a known limitation, I guess, of the CNMF method in general. I think our decision to use CNMF was because it's been a standard method in the field, but I agree that it's possible that some of the programs that are maybe less dominant because of lower expression or low variability might be best. Okay, so with that, we move on to the next section, which is looking at how do AI agents integrate into computational biology workflows. And for this specific case, we decided to look at the targeted hypothesis exploration part, because I think a lot of the human expertise and time and effort actually goes into not just running the pipeline, but actually specifically following up on one line of questioning that seems to be the most interesting or most relevant to that specific data set that's being analyzed. So to do this, I want to first define the different kinds of human-AI interaction modes that we consider. So the first mode is human in the loop. So human in the loop means that the human is actively guiding and shaping the AI's agent's reasoning. During execution, it would provide input. They could give more feedback about how the agent is doing the steps, could ask the agent to repeat the steps if something's wrong, etc. So very involved. Next is human on the loop. So you can think of it as kind of like a security guard. So it's very passive. The human is monitoring the AI system's actions. They're not necessarily trying to change the direction of reasoning, but if the agent is making a mistake, it could be something as simple as it saved the file in the wrong place. It's supposed to save an image that's saved in the wrong place. You can correct it. Or it could be that the agent ran something, but ran it with the wrong parameters. So that could also be corrected. So the reasoning aspect of it is not changing, but any mistakes that the agent has been making, the human is able to correct that. And so that's the way in which human on the loop feedback system is working. And then we have the autonomous system in which there's no human involved. The agent is operating independently and making its own decisions, taking its own actions. So for this specific part of the study, we chose to look at 3 different data sets that were recently published in the past 1 year. The first was the liposarcoma data set, in which the primary question was, what are they had liposarcoma samples of dedifferentiated and well-differentiated patients. And the most prominent question in the paper was, what are the transcriptomic and epigenetic differences between these 2 subtypes? The next data set that we looked at was esophageal adenocarcinoma data set, in which the primary question was about disease progression and which of the malignant cell programs are associated with the disease progression in this specific cancer type. And finally, we looked at a breast cancer data set, in which the specific question was therapy resistance in HR-positive breast cancer. We invited the original first authors of these publications to actually also join our study in order to be able to evaluate the response of these agents, responses of human plus AI final that happened, and give us more better insight into what's really missing and what's really happening. So in order to give a little bit more detail about the experiment design, the task is again related to CNMF, but a little more specific, concretely defined, because in the initial few runs of this experiment, we found that humans actually have very different ways of thinking about things. And so if you give a very broad task, they would go in very different directions because they interpret it very differently. And so we defined goals, but we did not define the method to achieve that goal. So we asked the 4 different subtasks to be completed. First is the CNMF program annotation. Next is the hypothesis formulation, which basically means that related to whatever the question of that data set was, which of the CNMF programs seems to be the most relevant. Third is regulatory driver analysis, which basically is trying to get at that, okay, you've identified 1 or 2 of the CNMF programs that are most related to the task. Can you identify what's a regulatory mechanism that's in play to bring about whatever the CNMF program is trying to do? And fourth is the biological interpretation of like trying to combine all of the things together and making a coherent interpretation of what's happening in this disease context in this data set related to this question. So this task is the same for all of the 3 data sets that we have. And we have different humans of different levels of expertise participate in this experiment. So the dark blue color here is indicating the first author publication or first authors of the publication, or in one case, we also had a single cell plus cancer biology expert. So we thought it would be a nice way to compare. The middle is people who have generally single cell experience. They've done single cell analyses, but they're not specifically familiar with this data set, or they haven't like worked with this data set, and they haven't necessarily participated. And then the gray ones are people who have general computational biology experience. They've never worked with single cell. They might have worked with bulk RNA-seq, they might have worked with other genomics data sets, but not specifically single cell. And we ask, and then of course there's the AI agent that's running in the autonomous mode here. The sign here indicates that the AI agent was actually blocked from accessing any of these publications, because in this case we had the web tool configured, and so we were finding that the agent would actually sometimes find the paper and would try to cheat. And so we actually we blocked the agent from accessing certain URLs all related to the bioarchive versions of the paper, any other versions of the paper that were available on any other site, the GitHub repos, and so on. And all of these humans participate in the experiment in 2 modes. So the HITL mode is the human in the loop, in which the people are more involved. And the HOTL mode is human on the loop, in which they're passively observing and correcting the agent, but not very involved. As a result, for each of these each of these participants, we have 2 sets of final responses related to them solving the task with the help of an AI agent. And the black one here is just indicating the autonomous mode that we run the AI agent in. After we have collected all of these, we then go back to the experts and we ask them to evaluate all of these runs with very specific questions. Note that when we go back to the experts for the blinded review, we actually remove their own runs to make sure that, because they obviously remember their own runs, to make sure that it's blinded and proper way of reviewing. So if you just generally look at what humans were doing with these agents or what these agents were doing, this is actually even true for all the other setups that we had. Agents are spending a lot of time trying to explore the data and actually understand the context. On the x-axis here is the number of steps, basically. So the agent has been doing many steps of exploration compared to, let's say, domain-specific bioinformatic analyses. It would also spend a lot of time trying to interpret the results. And as you can see, like there's a number of categories that we have here. When you look at specifically user feedback, which is only present in this section of the study where we have human plus AI things, we were curious to see how humans were interacting with the agent in the HOTL and the HITL mode. So human on the loop, where they were passively interacting, they were giving very little method direction. Like they would maybe correct something like a parameter that was done wrong or something like that. They were not necessarily, but it was much less. That drastically increases in the HITL mode, in which we see methods not being just asked to change things in the methods, but also trying out other different methods that haven't been done by the agent by default. And then there are some other different kinds of things that these humans were asking the agents to do. We wanted to do one sanity check when we were actually looking at the blinded review, when we gave the blinded reviews to the experts. We wanted to make sure that there are no biases that they have that could affect how they're reviewing. So sometimes people are more comfortable with AI and they trust AI more, so they might be favoring AI. It could also be the other way around. And we didn't really have a good way of knowing who lies where in that AI trust spectrum. And so we wanted to make sure that they're not able to identify which runs are actually human on the loop, which runs are human in the loop. And they could also not identify like the autonomous runs from each other. But I'm just showing here for human on the loop and human in the loop because I think it's very, very interesting to see. The arrows here indicate the accuracy specifically. So for the actual human on the loop task, the reviewers were able to guess human on the loop only 29% of the times. 53% of the times, they thought that it's actually human in the loop. And similarly, we see the same thing happen for the human in the loop part, in which humans, basically to say that they're not able to guess which is which, and they're not able to, hopefully not able to be influenced by how much AI was autonomously doing the things and how much human was involved. So when we look at the scope, so for each of the tasks that for the reviewers that we had, we asked them to rate on a scale of 1 to 4, 1 being the best and 4 being the worst. We find that, sorry, I think it's 1 being the worst and 4 being the best. And so when we average the scores across different kinds of tasks, here's T1 is CNMF program annotation, and T2 is hypothesis formulation, in which you're selecting the CNMF programs related to that specific datasets question. We find that these are relatively simpler, and so the agents are actually doing autonomously pretty well. Adding humans in the loop was not necessarily very helpful when you average all this across different datasets. But for some of the more difficult tasks, which are identifying the regulatory mechanism or making a coherent sense of like what's really happening in the data set, like the biological interpretation part, we see that agents are not necessarily doing better, but they're somewhat comparable to different kinds of human interactions. This is to say that for some of the tasks where things are somewhat standard and humans do it pretty much as a combination of 4 or 5 different ways, agents are able to get to that stage significantly okay. But for some of the things which are more open-ended, like regulatory driver analysis or biological interpretation, agents are still similar-ish, and they're not necessarily any better than a lot of the humans. I would note that we only had 12 humans participate in this, and so of course there's no statistical result that we want to claim here. We are just trying to claim trends. But with that in mind, it still seems that there are a lot of similarities in how autonomously agents are able to perform versus they're able to perform in tandem with humans. So we asked, one of the questions that we asked the experts was to identify if for any of the tasks, any of the specific outputs that they had in the blinded set of review was better than their own paper that they had. So they had already published, they already knew one of the things that happened that could be a plausible interpretation of that data set. And we showed them these 7 to 9 different other outputs, and we asked if for any of the tasks, one of them was better. And we found that a lot of the times humans were identifying outputs from tasks which were run by CB here stands for computational biology experience, and SC stands for single cell specific experience. And we find that a lot of the times they were actually finding the HOTL tasks to be pretty good or better than their own versions, equal or better than their own versions. And in 3 of the cases, actually even autonomous to be really good. Interestingly, for human in the loop, when the humans were involved a lot, they weren't necessarily always scoring top on the rate at which, on the scale at which the experts were reviewing. Some other qualitative observations that we had, which again, limited in statistical power, but were clear trends. So we find overall that more human involvement doesn't necessarily lead to better outcomes compared to human on the loop, so which is passive human involvement. And when we talk to people, I think one of the things that came up was that humans probably need some time to adapt to the working with the AI systems. And if they've not already worked with the AI systems, which was the case for all the participants that we had, like they had used obviously LLMs like ChatGPT, but they hadn't worked with AI agent in their workflow. They need to adapt working with it to actually be able to derive benefits. More human involvement we noticed was leading almost always to lower data exploration. So the agents initially would explore our data, but sometimes because humans have already looked at the data and they might be faster than these agents, they would have prematurely asked the agents, not prematurely, but they would have asked the agents to do something very specific and not allow the agent necessarily all the time that it wanted to explore the data. And that sometimes we felt was limiting the AI agent's understanding of the data set. And so its limitations on what it was able to do in the way it was able to do without as much involvement. And so I guess we come at the question of where are we, and where, like, overall, what's the broader takeaway of like where the field is going. It's still very early and it still keeps always changing with every new development, as we all discussed after the primer. But we think that AI agents are performing reasonably well on pipeline-driven tasks when the dataset complexity is low. So for example, in some of the benchmark papers that you will see, they have benchmarked on PBMC. PBMC is a simpler dataset compared to the cancer datasets that we had with multiple patients, single-cell RNA, single-cell ATAC modalities, which leads to more complex workflows. And if you would actually notice, none of the papers that get published are following necessarily the exact standard ScanPy pipeline that exists. People always have some modifications based on their dataset. The agents were actually almost always exactly following what the ScanPy recommended pipeline was. And that, I think, is what is a nuance that leads to a lot of difference between high complexity and low complexity datasets, and probably something that humans are really good at identifying, but the agents are not there yet. We find that agents also, like from the analysis, they show mixed success at prioritization. Some of the times they're prioritizing based on broader programs like EMT, but some of the times they're very specifically looking at like a program that definitely is more targeted, and they're able to prioritize that better too. It's unclear at the moment with current analysis, without looking at the model weights, how the prioritization is necessarily working. We believe that it could be a combination of task complexity and simplicity of the biological question, but it's still early, and I think more explanation on this needs to be done. Humans, irrespective of domain knowledge, whether they were computational biologists, whether they were single cell specific experienced people, or both of them did not necessarily show immediate benefit from human AI collaboration, and we are interested in further exploring what happens if you actually let them use AI for some time and then try to again see if they are deriving benefit or not. And finally, of course, as there's more new models coming every day, the model capabilities are changing a lot. It's possible that what we see with Opus 4.6 is some of these things may hold true in other models, some of these may be different. And so there's a need for constantly evaluating your own workflow as you are building these agents and as you're switching between models to make sure that you're not making assumptions based on a model that you battle tested 4 months, and you know that its weakness is A, B, C, and you always fix that. And when you go to a different model, it may have a different weakness. It may be doing great at A, B, C, but unless you are as careful as you were with the first one, you may not actually necessarily be able to capture or catch the smaller subtleties of what the agents are missing. With these, I would like to thank Ellie, who's been the mentor on this project, and Jihe for working alongside to wrap up some of this work, which we hope to preprint soon. And all of the co-authors who've been incredible thought partners in designing of different kinds of evaluations, which get at the question of not just pipelines, but beyond pipelines, what we as computational biologists do. Each and everyone on this paper is actually a computational biologist, and so they work in some in single cell, some in genomics, some in bulk RNA sequence. So they've brought in their own expertise into helping design the study, and very grateful to all of their support, and happy to take any questions.
So that was a great talk and a real stress test of like something a computational biologist would actually do. So awesome, thank you. So what are like the prospects for these other approaches where they use like these RAG approaches where it's like, I don't know, you give the model as well, like here's the 10 best single cell RNA-seq cancer publications, look at those methods and draw from that rather than ScanPy, or here's an immunology and an oncology textbook. They're like the standards in the field, draw from that. Like how much of that do you think is going to help, or is it not needed?
That's a great question. I think it would definitely help if the dataset characteristics are similar between the publications that are part of the RAG database versus what you're actually analyzing. So I think there is a possibility that it would help, but then you would have to probably select the RAG database to consist of publications that are more aligned. And I think it's definitely possible. One of the things that I didn't touch on a lot, which I think also is related to your question, is when we design these tools, we actually, it's nothing but a function. So if you specifically know that in cancers, these are specific things that almost always are different than how ScanPy does it, you would probably want to encode that knowledge within the tool. And we found that AI agents some of the times don't really use the tool. So maybe like just prompt it to use the tool to make sure that it's like actually reliable and actually giving you what you want. But I definitely think that RAG systems can help boost the performance.
Hi, great talk. That was really interesting. I was actually really curious about the first part where you were talking about the cell type annotation, the multi-omic pipeline. Did you ask the agent to iterate on the QC steps or no? Because would it have worked better to maybe do that if you wanted to actually iterate on QC?
That's a great question. So I think we as a team went back and forth on how prescriptive we wanted the tasks to be. And we wanted to capture I think as was alluded to in the primer, there's a lot of benefit in actually building a very robust scaffolding around the agent. So the system prompt that you give the agent can contain very much a lot of details about exactly how to do what to do. And it's definitely possible to use that to bridge the knowledge gap. And we in the study wanted to test what knowledge the agent inherently has. So we didn't really ask it to iterate on that workflow. But it was interesting to see that it iterated on the cell type annotations on its own, but it did not really iterate on the pipeline running step. But I definitely think that if you actually specifically ask it to iterate, it will definitely iterate.
Because I was also curious, you showed like the area under the curve plots, but or numbers, but which cells were used in the end? Did it use the same cells as, like, did it come up with the same cells after QC as the annotations? And then are you just looking at the cluster annotations, or is it a per cell annotation?
This is a per cell annotation. So,
but then was the data constrained by a cell type label per cluster?
That's an excellent question, actually. And we had a lot of, we had to do a lot to actually make sure that we are able to match the cell types properly, because we didn't provide the model that these are the cell types you have to find. The model came up with the cell type annotation. So sometimes, for example, the dataset would contain CD8 cytotoxic T cells as the label, and the model labeled it as a T cell. But because we hadn't really specified the model in how much depth you should go, we didn't really want to penalize it necessarily. So what we did was we took the original author annotations, we created a hierarchy from those of like, if there is immune cells, then immune, then T cell, then CD8, and we tried to match the annotation to the closest leaf.
Yeah.
And so it doesn't always get to the most fine-grained annotations. You're totally correct in that, and it's a difficult to benchmark task as well because we're not constraining it. But definitely, like, the agent was doing a lot of back and forth and trying to gets at some of the broader things.
It's just the harder problem is the more specific problem than the broader categories. But yeah, this is great. Thank you.
Yes, thanks. This is a great talk. My question is about the last part when you're evaluating the human in the loop, human on the loop, and the autonomous mode. And to do that, you mentioned you have to block the AI from accessing the original publication that contain your benchmarking data. I'm just curious, how did you do that? In my naive thinking, like, AI already seen this data if those are public information.
That's a great point. So all of these papers were actually published pretty recently, and that's why we also selected these papers and invited the authors on it. The bioarchive versions were still present before the original publication. So it's possible that they might have been part of some of the training that the model had undergone. We did not find that those bioarchive versions necessarily had all of the information in the text. Like, the information was present as supplementary tables, which would have like, the model trainers would have had to download. I'm not saying that's not possible, they didn't download, but it's less likely. So we tried to make sure that that doesn't, like, it's hard to build a complete wall, but we tried our best to do it. In terms of how did we block the access, so we changed the way that the function for web search was written. So anytime the model, so what happens inside of the tool, the model doesn't see. The model only sees the output that the tool gives. So you can, in the code, you can actually do anything. So we let like, let's say that I'm using one of the packages to pull all of the PubMed IDs for a related search. We make sure that the PubMed ID for our specific dataset was removed from that list before that list was returned to the model. So the model never saw the PubMed ID and so could never search the PubMed ID. Or the model never saw the bioarchive link, so could never actually look at the bioarchive link and like download the article or anything.
Or do you have a question? Okay. Hi, we have a question from Zoom. AlphaFold 2 uses transformer architecture and estimates the correctness of the 3D protein structure it has produced to determine how reliable its prediction is. Gen AI systems predict the next token or pixel. Is there a measure of correctness for Gen AI systems?
I think when you think about measure of correctness, it's probably different than the way we think about proteins. And that's why there are different kinds of evaluation methods that people are using. Also, in transformers, you're able to also look at the weights, and this is like a black box. So I think the field of evaluations of Gen AI systems is actively evolving. People are still figuring out the best methods to probe this black box system and try to get insights about what works, what doesn't work, and constantly keep updating that knowledge as the models evolve. So I don't necessarily have a good answer to this is the exact method, but it's an active area of research. Thank you.
Thank you so much again for the talk. I wanted to ask, to me, this essentially is a sort of one-shot human versus one-shot LLM comparison, especially in the second half. But ultimately, especially for the more complex analyses, even if you were to get 10 humans to do the exact same data set analysis, you might get 10 different answers. And similarly, if you get 10 LLMs doing it, you're going to get 10 different answers. And so I wonder, I mean, what, in some ways ultimately, when we're looking at these comparisons, we should probably, be helpful to see this as a distributional distance in some ways, and also to think about this as like are the answers that the LLM is getting something that actually human could also be getting that, as opposed to just, oh, it's different, therefore it's wrong. What are your thoughts on this?
Are you talking about the human AI part of the paper?
Well, both I guess the first half was kind of the CNMF comparison, right? Mm-hmm. The second half is human in the loop, autonomous, right? But I think both stand. I guess the second one is multi-step, more complex. But even with the first one, it's like, how much variability do you have within humans? How much variability do you have within LLMs? And then you can make that comparison, as opposed to saying one and one, and then therefore, humans are, this specific result, it must be the correct one, but actually there are probably other ones as well.
That's a great question. So for the human plus AI part, we actually did not match exactly because our evaluations were done by a human. So the human already made judgment calls of we were not saying that if the AI agent came to this exact same hypothesis that the human had come to in the paper, that's the correct one. We actually instructed the reviewers to make sure that they're open to possibilities and are evaluating not just what the final answer is, but actually the method through which the answer was attained. And if that seems to be defensible in justifying whatever the final answer was, so that part of it is not exactly matching. As for the first part where we have cell type annotations, it's definitely possible that a different human might label some of the cells differently based on how they run the pipeline, which is also the reason why we chose HTAN for that, because there's just there's a lot of papers which publish single cell multiome, and everyone does a different way. HTAN specifically they had a very detailed documentation of how the samples had to be processed experimentally and computationally. And so we felt that even if different humans were doing different cohorts, or if they might still be different, but they would still be within some distance of each other, that it can be treated as somewhat reliable, because HTAN actually has like these working groups and they debate a lot on how exactly they want to analyze the bulk of it. And so I think that was our thought, but it's again like totally possible that maybe if you or I analyze it, we come up with slightly different annotations for some of the specifically like more nitty-gritty details or like specific cell types.
Yeah. There's another question from Zoom. How do you see AI in the loop where humans lead the analysis and call up AI when they need as an alternative? When they need it as an alternative strategy to human in the loop based on your results. So instead of humans learning how to use AI, should AI learn how to work with humans?
That's a great question. I think the model developers are trying to make the AI learn how to work with humans because they're training a lot using RLHF, which is reinforcement learning with human feedback to some extent. But I think the core of the question here is that humans are very different in how they work with AI. They have very different trust levels, which affect how much what kind of interactions they do with AI. And so every person's learning curve would actually be very, very different. And it might take me a month to get more comfortable using AI and understanding what it does versus someone else who might take a week maybe. So I think we can't really, I'm not sure if we are able to control much of how the model developers are doing controlling AI to make sure it works well with humans. But I think we definitely need to make an effort, and it wouldn't be like a one-shot immediate thing.
Thank you so much. And yeah, that'll be all the questions for now, but we'll stick around and the speakers will stick around for a little bit if you want to ask more questions.